Hello everyone, a very good evening to everyone. Okay, so let us discuss about the cell cycle. Okay, so in cell cycle we have to study two things, that is mitosis and meiosis. So let us look at them one by one. Firstly, let's read about mitosis. So this is a complete cell cycle or the mitotic cycle which we are discussing here. It consists of G1S, G2M, cytokinases and G0. We'll begin with G1. G1 is the gap phase 1. In this, the cell is metabolically active but doesn't grow. During the transition to S phase, the cell increases in size due to accumulation of products. The nucleus in the G1 phase has 46 chromosomes. Here, the chromosome looks like this. During the S phase, when duplication of the chromosome or the DNA material occurs, then the chromosome looks like this, but this is also one chromosome and this is also one chromosome. Here there are two chromatids. Here they are more than two chromatids. How many chromatids are there? There are 4. Okay, so during the S phase we have 46 of these. The number of chromosomes is same, but DNA content doubles during the S phase. The next is about the metaphase. The next is about the M phase, which is prophase, metaphase, anaphase and telophase. Before the M phase, the protein synthesis takes place in the G2 phase. Now during the M phase, we have to remember it as PMAT, prophase, metaphase, anaphase and telophase. Alright, so what happens in prophase is untangling chromosome material and the dissolution of nuclear membrane. The next, in the cell, okay, this is in the nucleus and this is in the cell. In the cell, the duplicated centriole moves towards the peripheries, like opposite to each other. One centriole will become here and one centriole will come here, just as in next diagram. Now, after these centrioles are at their place, they give out spindle fibers which attach to the kinetochore region of the chromosome. Since we know that we have 46 chromosomes at the moment, each of them will be separated and they'll move towards the opposite pole in the next form. Here all are arranged in the center, called as the metaphagic plate. The next is anaphase. The spindle fiber splits it up and the two parts move towards the two opposite poles. This is going to move upward and this is going to move downward. Hence, in the last telophase, this here is telophase. In the telophase, the nuclear membrane reappears and we can see that we have two nuclei of 46 chromosomes each. The furrow begins to appear. The cytokinases is done. After the cytokinases, we can see that there are two cells. There are two cells with two different nuclei. Now each of them, if I say about it, each of them will have 46 chromosomes, right? 46 chromosomes. Okay, so we are done with the mitosis. Now let us look at meiosis. So meiosis is divided into meiosis 1 and meiosis 2. In meiosis 1, we have prophase 1, metaphase 1, anaphase 1 and telophase 1. The prophase 1 is further divided into leptothene, zygotene, pectine, diplothene and dikinases. Starting with the leptothene, the compaction of chromosome occurs. In the second one, that is zygotene, the zygote formation or the pairing of homologous chromosomes occur. We know that there are 46 chromosomes, each occur in pairs, so we have 23 pair of homologous chromosomes. Homologous chromosome means that this is the chromosome 1 from female parent, and this is chromosome 1 from male parent, female parent and male parent. Okay, so we have 23 such pairs. In zygotine, what occurs is that the synaptonemal complex formation occurs at the synapses. Okay, homologous chromosome pairing occurs. Okay, in the next, that is pegitine, the chromosome here are visible as tetrads. This is how a tetrad looks like. There is crossing over between the non-sister chromatids. Okay, now what occurs in diploidine is that dissolution of the synaptonemal complex. It means that after the crossing over, these are to be separated now. Can you see this X-shaped chiasmata? This is the region of crossing over. Now you can see this is the A chromosome and this is the B chromosome. These are the pair of chromosome number 1. Let's say, okay, it can be any number from 1 to 23, right? So let's say that this is the pair of chromosome number 1. So you can see that this genes, these are some areas which I have marked. Let us take them as alleles of a gene. Alright. After the crossing over has been done, you can see that these alleles are shifted to different chromosomes. Right? Here, if I say about this pink region, this was present on a chromatid of B. But here it is present on A. So this is what crossing over does to the chromosomes. The next, nuclear membrane dissolution occurs. And this last part is known as the diakinesis. In diakinesis, crossing over is done and terminalization of chiasmata occurs. Also, the nuclear membrane dissolution occurs. After this, we come to metaphase. This particular is metaphase. In metaphase, as we know that the chromosome, these homologous pairs will arrange themselves in the metaphagic plate. In mitosis, we would have a long metaphagic plate because there were 46 chromosomes arranged this time. But here, since they occur one above the other, so here these will be short and we will have 23. Understood? This is the metaphagic plate of meiosis 1, and this is the metaphagic plate of mitosis. Alright. Next up, what happens is appearance of nuclear membrane. After metaphase, we have telophase. So, the chromosomes are now moved apart in the anaphase, right? This one is the anaphase. These will begin moving towards the opposite pole, that is in the anaphase. Let me mention it here so that it is clear. Anaphase 1, movement towards ends, right? Then appearance of nuclear membrane. Now the cell furrow will develop and cytokinesis will occur. Alright. After cytokinesis, we will have two cells, right? Each has 23 and 23 chromosomes. Okay. So now what occurs now is meiosis 2, and meiosis 2 is same as mitosis. Now you can see that each of these, each has 23 pair, right? So now it will be similar to mitosis. For example, if I tell you that prophase, metaphase, anaphase and telophase, if I have these 23 chromosomes, what will happen in metaphase? These 23 chromosomes will lie like this. Like I have drawn 2, there will be 23. Okay. In PMAT, this is one chromosome, right, which this cell has. There will be 23. In metaphase, these will be arranged like this, and these will be attached to the centriole, right, from both the sides. Now in the anaphase, it will take it away just like this, just like it did in mitosis, which we have already studied. These will move apart and then these will lodge in separate nuclei and the cell will begin to furrow, right? And then you will have two cells with two different nuclei, and each of these nuclei will have as such 23 chromosomes, right? Okay, well and good. Now we come to the end. We have discussed mitosis, meiosis 1 and meiosis 2. This is what you need to understand. Thank you for your patient listening. Good luck.